So we have the digestive system. We're going to go through just the, I'll go through just the list of, like the study guide list for you guys, and then we can hit other images if we want to see things more clearly. So for this first part, you should know the parts of the peritoneum. So what are the two parts of the peritoneum? Parietal and visceral. Parietal is going to be the bubble around the intestines. Visceral is going to actually be touching the surface of the peritoneum. What are the four main layers of the GI tract? The innermost layer touching food is going to be mucosa, and then beyond that it's going to be the submucosa. Then beyond that, muscularis externa, and then beyond that, the outermost layer with connective tissue is the serosa. That's right. So the functions, really, the mucosa layer is going to be what we interface with the food. So different parts is going to have a different role. So you want to think of, like, in the esophagus, it's more about protection. So the epithelial tissue in that region is going to be more about abrasion. So it's going to be stratified squamous epithelial tissue. Where in the stomach, we're more about secreting the components that's going to make pepsin, digest proteins. So we have to have a combination of not only making these agents, but then protecting ourselves from our own digestion. That would be the goblet cells. So there's going to be different cell types there for that purpose. Then the duodenum, we have to have a mixture. We want to protect ourselves from acidic bile coming in, but we also then want to provide the opportunity for absorptions. We have villi, we've got the simple columnar cells, but we also have those bicarbonate cells to create, to buffer the acidity. And then, so throughout the whole GI tract, you're going to have some variations. And then you've got the submucosa that's going to be bringing in vessels, or a lot of the vessels are housed with connective tissue, whereas the muscularis externa is going to be doing its contractions to move food along. The autonomic nervous system role in GI function, I really don't have any detail in terms of the control outside of, I want you to realize that the GI function is primarily going to be parasympathetic. It's not evenly balanced. This is one of those areas where the sympathetic nervous system really doesn't do much outside of just shut it down when you're scared or running. Like, pretty much divert flow elsewhere, digestion can go on hold. But, over like almost 90%, 70-90% of the action of the digestion and moving food along is really a parasympathetic driven thing. So I just wanted to kind of point that out. Things aren't always going to be even. You should know about the structure of teeth. So we talked about the neck, the crown, the roots. You should know the anatomy of a tooth as far as the enamel, the dentin, the pulp cavity, the root canals. You should know the edges have cementum and the periodontal ligaments, and the gums are going to be the gingiva. And so it's going to sit down in your teeth. The gingiva is going to go around and protect it. You should know the types of teeth and the number of each, and you should know the total number of teeth. Then the location of the salivary glands. You should know the parotids are near the masseter muscle on the ramus of the mandible. You get submandibular salivary glands, which are under the mandible, and the sublingual, which is under the tongue. You should know what they secrete. The two main secretions that I want you to be aware of, aside from water, are going to be salivary amylase that digests carbohydrates, initiates the carbohydrate digestion. And then we have the bicarbonate, which will neutralize any acidic food coming into your mouth. The histology of the salivary glands are such they look like they have like dark and light acini. These are like the little glandular components. The submandibular gland is the one that has the most evenly balanced number of the dark and light. So if you were to see them on a practical exam, I would utilize the submandibular one. One concern that you should have is the submandibular often looks like the pancreas, so you want to make sure you can discern the difference between the two. The components of saliva really is mostly water, mucus, that salivary amylase, and the bicarbonate. Any questions on this or any areas that we want to kind of go into a little more detail? Okay. I'm going to jump forward to the next section. So part 2 for the esophagus and stomach, you want to look at or be familiar with the esophagus structures and histology. So that's structures really being maybe the sphincter muscles. They're not as obvious structure. The histology is going to be the one that I'd really focus a little bit more on here, where we had, for instance, that last slide in that little quickie quiz we did. Longer than a quickie quiz, I guess. But you have the mucosa layers, you're going to have stratified squamous epithelial tissue, you're going to have the submucosa, you're going to have the circular and longitudinal muscle that make up the muscularis externa. So you'll just, if you see a histology slide of the esophagus, you can tell that it is that generally because you almost always see it curved like a tube. A lot of the other areas we're looking a little bit deeper and more high-powered. So the esophagus, generally we don't look at it so high-powered, but the inner mucosa part looks unique to the other areas because it is that stratified squamous. The fresh cells at that bottom layer is going to be dark purple and it usually fades, or a dark pink. But so that kind of gives it a unique look too. The stomach structures and histology, there's a lot more anatomy going on with the stomach. So we had the cardia region, we had the fundus, you had the body, pyloric region, you had the pyloric sphincter, you've got rugae inside. So there's a lot more features of the stomach. You should know the lesser omentum hangs up, goes upward from the lesser curvature of the stomach, and the greater omentum hangs below. So the way it lies in the GI tract, once you open the parietal peritoneum, you'll see the greater omentum laying down over the intestines. You'll see the surface of the stomach, and then you'll see the lesser omentum, but everything else is pretty much covered up. The histology of the stomach, we know that the mucosa layer is going to have those deep gastric pits. So at the top of the pits, the closest into the lumen is going to be our goblet cells that make mucus. Down in the middle third of the pits, we're going to have the parietal cells that secrete hydrochloric acid and intrinsic factor. And then at the bottom is going to be the chief cells that secrete pepsinogen. You should know that the parietal cells that secrete the intrinsic factor and hydrochloric acid, what the role of each one of those things are. Hydrochloric acid is used to convert pepsinogen into pepsin and then further drop the pH in the stomach so that it can activate the pepsin to digest proteins. The intrinsic factor binds to vitamin B12 so that that combination can be absorbed in the ileum. The stomach is the only place that has 3 layers in their muscularis mucosa. In their muscularis externa. So the the extra layer is called the what layer? Destiny? The oblique layer. Yes. So it's called the oblique layer. It's the only one that has that extra layer, and it's going to be just adjacent to the submucosa. Chemical digestion of proteins, you should know that. We just sort of mentioned that when I was talking about the stomach mucosa. That's where you have chief cells secreting pepsinogen. You have parietal cells secreting hydrochloric acid. They combine to form pepsin. Additional hydrochloric acid drops the pH so that now that pepsin becomes active and it starts to break those peptide bonds. Who can tell me what the role of mucus in the stomach is? Protects ourselves from our own activated pepsin from getting into the wall of our stomach. So if you don't have enough mucus in your stomach, you're going to have an ulcer because you're going to digest yourself. Small intestine. The 3 regions of the small intestine are the duodenum, jejunum, and ileum. The duodenum secretes bicarbonate. The duodenum receives bile from the gallbladder. The duodenum receives pancreatic juices, which are just pancreatic enzymes that can digest fat, which would be pancreatic lipase. The duodenum receives trypsin from the pancreas, which digests proteins at a neutral pH, and the duodenum receives pancreatic amylase, which digests carbohydrates from the pancreas. The duodenum has the Brunner's glands in the submucosa that secrete the bicarbonate, which buffers the acidic chyme from the stomach. So that is a very unique and distinct histological feature. Then there is the jejunum. Major job is for absorption. So we want to have long, slender villi, many villi, very long to increase surface area. The ileum has shorter villi. The ileum is where we absorb vitamin B12 as long as intrinsic factor is bound to it. The ileum has the unique histological feature of the Peyer's patches. The end of the ileum is where we have the ileocecal valve. Once that ileocecal valve is open, the food from the small intestine will go into the cecum, which is the start of the large intestine. The large intestine has a fair amount of anatomy with it. So you should know the cecum, the appendix hangs off of the cecum. We have the ascending colon going up. We have the hepatic flexure on the right side. We go to the transverse colon, and then we go to the splenic flexure on the left side. Then there is the descending colon, sigmoid colon, and the rectum. The other unique feature of the large intestine is the stripe that you can see on the surface, known as Taenia coli. And then the stripe, the contraction of that creates these pouches known as haustra. Inside the large intestine on the histology, you just really see from top to bottom of the mucosa, lots of goblet cells, just a lot of white circles. So that's really the primary unique feature that you're going to be looking for to identify a colon slide or a large intestine slide. So the accessory glands and digestion, they're listed differently just because it's not really part of the tube that food has gone in. So now we're sort of, these are things that are contributing to the tube, but the food isn't going in these things. So the pancreas, we've mentioned already, is off to the side adjacent to the duodenum, is going to send secretions that will cover digestion of fats, proteins, and carbohydrates. And there's lots of them, but I'm just going to pick one for each one of those things. So if I were to say, you know, the pancreas releases what to digest fat, it would be pancreatic lipase. And one of the many things that it uses to digest proteins, but we'll just name one, will be trypsin. Trypsin is made by the pancreas, released in the duodenum, and it breaks down peptide bonds at a neutral pH. Whereas the stomach made pepsin, which digested proteins at a very low pH. So the pancreas then also makes pancreatic amylase, which digests carbohydrates. The histology of the pancreas is that it looks like more of one continuous, the cells in it to me looks like one continuous carpet with just some lighter regions that are the islet of Langerhans, and the darker regions are going to be the acini. It's less divided up, I think, visually compared to the submandibular salivary gland, because that's the one that people confuse it with. As far as the liver, the function of the liver is detoxification. We make components of we create proteins from our liver. We create fat from our liver. We can create carbs from our liver. We create plasma proteins that's going to circulate around, clotting proteins. So the liver creates a lot of things. It detoxifies and removes waste as well. It forms bile. Bile has a lot of the waste, like old cholesterol, things that we want to just dump out and excrete and remove from our body. But we also utilize bile to emulsify fat. That means mechanically separate fat into smaller droplets. In the liver, we bring in blood to the liver. There are two vessels that bring blood to the liver. One is the hepatic artery, the other is the hepatic portal vein. That's the blood that would have waste that the liver would then remove. So the clean blood arrives in the middle of these lobules into the central vein. The central veins have the clean blood that then exit the liver via the hepatic vein. The waste leaves the liver via bile ducts. The bile then leaves the liver as a whole via the common hepatic duct, and then goes into the cystic duct to be stored into the gallbladder. And then if it's going to leave and go to the small intestine, it goes down the common bile duct. The digestion and absorption process of carbohydrates, fats, and proteins. So if we're going to start with carbohydrates, it starts with your mouth. We're going to chew it. That's mechanically digest. We add in some salivary amylase, and we start the carbohydrate digestion. That gets bumped down into our stomach. There's no more carbohydrate digestion going on outside of just mechanically mushing it around a little bit more. Then we finally get it into the duodenum, where we add pancreatic amylase, which is going to digest carbohydrates. Proteins chew in the mouth. Then we drop it down, so it's only mechanical digestion in the mouth. The first chemical digestion from proteins happen in the stomach from pepsin with the low pH. And then we finish the digestion of proteins in the duodenum. Then fat, we just don't even think about any of the other parts until it gets to the duodenum. We break it down, mechanically digest it, and emulsify it by bile in the duodenum, and then we digest it by pancreatic lipase in the duodenum. The hormones of digestion, you have gastrin, which when protein arrives in the stomach, it tells the stomach to start churning, releasing the pepsinogen and all of that. The other two, secretin and cholecystokinin, are in the duodenum. They both tell the stomach to slow down. One does it because of acidity, tells bicarbonate to be released. The other, cholecystokinin, tells the stomach to slow down while it's also telling the gallbladder to release bile. Okay, so we'll start with our metabolism review. The 3 parts of metabolism. The majority is going to be in this first part. We'll do carbs, go through the whole pathway of the carbohydrates, just talk about how lipids, where they step in, and then what's going on with proteins. So glucose processing, who can tell me some terms for glucose processing? There's glycogenesis, it's making glucose. Then what's the term for breaking down glucose? Glycogenolysis. You're breaking down, or making down glycogen. Sorry, I termed that wrong. Let me go here. Yeah, glycogenesis is forming glycogen. Glycogenolysis is breaking down glycogen. So one is sending glycogen, forming glycogen in the form of temporary storage of energy. And then when you need more glucose later, then you're going to go through glycogenolysis to bring your glucose levels up. Gluconeogenesis happens when you create glucose through non-carb sources. What would be a byproduct of gluconeogenesis? Ketones. Excellent. So glycolysis. Where is glycolysis going to take place in a cell? Not the mitochondria, so that would put it out in the cytosol. Destiny's on it. So then, what does it produce? What do we get out of glycolysis? Get 2 ATP, 2 pyruvic acids, and 2 NADHs. Yep. So what happens to pyruvic acid in an aerobic state? Pyruvic acid turns into what? Acetyl-CoA. Maybe I should, I'll write that down. So we go pyruvic acid is going to become acetyl-CoA. Acetyl-CoA is going to go where? It's going to go into, and do a little dotted line. A little here, because we're now in the mitochondria. Okay, so it's going to go into the Krebs cycle. The Krebs cycle creates what? It is going to create, yes, carbon dioxide. That's sort of its waste product that we get from the Krebs cycle. And what else does it make? We get a couple of ATP that come out. That's kind of handy. We get then more that we're going to move along will be these more NADHs, FADH2. So we're going to leave that be since we're here at this carbohydrate. So as far as glycolysis goes, we got 2 ATP, we got 2 NADHs, which we'll also send over here and talk about it in a moment. Then we get the pyruvic acid. Pyruvic acid becomes acetyl-CoA so it can enter the mitochondria and enter this Krebs cycle. During anaerobic conditions, which really means we made a bunch of pyruvic acid, we turn it into acetyl-CoA, and we run it through the Krebs cycle, and we use oxygen later through the electron transport chain. This is still going, but shoving more acetyl-CoA, it can't handle anymore. It's just using it and burning it as quickly as it can. But our body needs even more energy. So we continue anaerobically by doing more glycolysis so we can get the 2 ATP out of glycolysis, but we're still making pyruvic acid, which is coming into acetyl-CoA and it backlogs. So, or because it's backlogging in its conversion to acetyl-CoA. So what does it become temporarily? Lactic acid. So then pyruvic acid, if it can't become acetyl-CoA, will come out and become lactic acid temporarily until the movement of acetyl-CoA into the Krebs cycle is now sufficient to be able to take this lactic acid and turn it into acetyl-CoA, and then we can eventually use it for energy. So the Krebs cycle, where it takes place, the byproducts and the end results. So where it takes place is in the mitochondria. A byproduct would be carbon dioxide. And the end results, as we get some ATP, we have the CO2, but we're getting NADH, FADH2. These guys are like tokens for more ATP. So they are tokens where the NADH is going to become 3 ATP. The FADH2 is going to become 2 ATP. And this process is via the electron transport chain. In order to utilize the electron transport chain and make these ATP, we're going to need oxygen. So O2 is required for this reaction. So we don't just get 3 ATP and 2 ATP total. It's for every NADH we get 3 ATP, and we get many of them. So we get, like, was it 10 NADHs, and you can add them up. And anyway, so you get lots of them. That's just what they ultimately become. So you'd multiply it by however many you get. There we go. We covered all those things. Any questions? Okay, we'll go to lipids. So you should know the types and structures of lipids. So lipids could be a steroid. It could be a triglyceride. We spend more time talking about the triglycerides because that's the main one we use for energy. But the steroids are cholesterol-based, and they're used to make hormones. It's also considered a lipid. Then we also have phospholipids, which are used to make our cell membranes. So these are things that our body inside actually uses fat for a purpose, for a good, not just for energy, like triglycerides. So how we process lipid processing, and we'll talk about the triglycerides, is if we have a triglyceride, we have this glycerol backbone, and then again with the fatty acid chains, 3 of them. We digested them as triglycerides, broke it apart, absorbed the individual components, and reformulated them back together for storage in our adipose tissue. When we want to go undergo lipolysis or lipolysis, we can break these bonds here, and then this glycerol backbone is actually going to go through glycolysis and end up being pyruvic acid. The fatty acid chains, they go through what's known as beta oxidation to break these long chains, which are carbon-linked chains, into carbon to carbon. So just a 2-carbon unit, which then can become acetyl-CoA, and we burn it through the Krebs cycle and the electron transport chain and redeem our ATP that way. So the energy that we get from the breakdown of triglycerides is enormous because of the long chains. If you think of a single triglyceride, how long each of these chains are. So just one of these carbon-to-carbon units makes almost as much as a single molecule of glucose, outside of the 2 ATP that glucose made from the cytosol. So there you can see how fat is an incredibly wonderful storage of energy and very, very helpful for people to transport their energy in times of uncertainty in terms of their food availability. What is ketogenesis? Formation of ketone bodies because you don't have carbohydrates to turn directly into glucose. The brain will only live off of glucose. So the body has to do some trickery and say, all right, I'm going to take the fat, turn it into glucose, but in doing so, ketones is this nasty byproduct that comes from that process. So it allows us, it's an adaptation that allows us to stay alive and keep our brain going, even if we're not eating carbohydrates. It's not a very efficient way to do things, but that is indeed a way that we can to help keep our brain survive. So ketogenesis is formation of ketones in the absence of carbohydrates because we have to make glucose from a non-carbohydrate source, which would be gluconeogenesis. Last section is going to be protein metabolism. This is pretty simple in that we digest proteins. You should know about digestion of proteins in the stomach and the intestines. I threw in the brush border enzymes because there are some enzymes that just cleave the 2 amino acids. It also does that actually for carbohydrates, breaks the last 2 little saccharides into little monosaccharides. But the main thing for proteins with regard to metabolism and energy is the process is known as deamination. You pop off an amine group. That doesn't really mean anything to you guys until you take chemistry. So it really is taking this protein molecule, and if you pop off the amine group, it's called deamination obviously, then the protein can come in, and once it's deaminated, will become acetyl-CoA, and we can then burn it. So protein can be a very useful form of energy if we're starving to death and we have no other food coming in. Obviously, protein is not a primary source of energy our body wants because it really means that we're breaking down something that we would otherwise use for a good thing in building up our body. So the main thing is deamination. You should know that there were 20, are now 21 amino acids, different types of amino acids. We did talk about essential, non-essential, and conditional, but you don't need to know that for the point of this test. It's just, I pointed them out. So you should know that there are 20, had been 20, currently 21 different types of amino acids. And if we're going to utilize an amino acid, or sorry, going to utilize protein for energy, it must be deaminated. But the main thing, whether you're using fats or carbs or proteins, is they're all going to become acetyl-CoA. That then funnels into the mitochondria for the Krebs cycle, which makes carbon dioxide. The tokens that we get, NADH, FADH2, go through the electron transport chain, which is where we utilize oxygen, and this is where we get the bulk of our ATP that we live off of. And that's your metabolism story. Any questions? All right, respiratory, get this done. We have the breaking up into 3 parts. One is really just the airway anatomy. So it's like the tubes that get the oxygen to the alveoli so we can exchange it, which is going to be part 2, where this, that's part 3. So the airway anatomy, is the tubes getting the air down into the alveoli. Part 2 is going to be overall structure of the lungs, that basic anatomy, and then how we breathe. And then 3 is going to be actually what we're going to do with the oxygen. Get it loaded onto the blood, get carbon dioxide released from the blood, and so on. That's where the actual exchange is going to take place. So for airway anatomy, you should know the regions and the functions. So let's start with air coming in. We'll talk about the nose. What is a unique feature of the nose, and what is the purpose of that feature? Nasal concha, we have 3 of them, superior, middle, and inferior. What do nasal concha do? Create air turbulence, excellent. So we want to create turbulence. When we create turbulence, we slow the velocity of air coming in down, and we want to slow it down so it hangs out in our nose for a little bit. For what 3 purposes? We want to warm it or equilibrate it to our body temperature, if you want to call it, be more technical. Then what? What? Oh, moisten, yeah, humidify. Humidify and then filter. Filter it with any of the mucus and the hair in there. So then it goes from the nasal cavity into the nasopharynx. Then it drops down to the oropharynx. And then it drops down to the laryngopharynx. And that point, it's going to go, if the epiglottis is open, into the larynx. Inside the larynx is where we house our vocal folds. We have the cartilage structures that are located within there. So we have obviously the epiglottis flap. We have the thyroid cartilage, cricoid cartilage on the back of that. We have the arytenoid cartilages with the corniculate sitting on top. I'm trying to think of back up in the pharynx is where we have a lot of all of our tonsils, which is our immune system guardian of any incoming any incoming air to our body to protect ourselves from any potential antigens. We go down from the thyroid cartilage, the cricoid cartilage, to the trachea. The lining of the trachea is made of pseudostratified columnar ciliated epithelial cells. We have mucus embedded within there, so the cilia and the mucus will trap additional airborne debris that made it past your nose, brings it up to the back of our throat. Obviously, either we expel it out or we end up swallowing it. So that's what's going on in the trachea. The trachea branches off into the right and left primary bronchus. The right primary bronchus is going to be a little straighter and pointing downward more. So if something were to make it past the larynx, it's more often not than trapped on the right side than it would be on the left. Every division of bronchioles beyond that is named for each division. So once you have the primary, then you had secondary, and it goes on from there until you finally go out to the terminal bronchioles, where on the histology, you start to lose smooth muscle and you lose some of the hyaline cartilage that's helping to keep that airway propped open. The ends, where you end up at the alveoli has is made of simple squamous epithelial tissue. It's flat and ideal for diffusion. So oxygen that's in the alveoli can move out easily to the blood. Carbon dioxide from the blood can move easily back into the alveoli to be gotten rid of from the body. Then we also have surfactant cells, which create surfactant, which type 2 cells, which create surfactant, which help reduce surface tension, so it prevents our alveoli from collapsing in on itself due to the water vapor. Back up to the vocal cord anatomy and muscles. You should know the vocal cord, cords, plural, are within the thyroid cartilage. They are attached in the front to the thyroid cartilage. On the back, the movable side, is the arytenoid cartilages. And so the muscles of our speaking is actually attached to the back side, moving our vocal folds together, pulled tighter, looser, and whatnot. So lung anatomy, breathing here, structure and histology of bronchioles. So the bronchioles, we have the lining is going to be pseudostratified columnar epithelial tissue. Beyond that, another important feature is the hyaline cartilage. It's C-shaped, so it doesn't go all the way around. It goes about 3/4 of the way, on most of it. And then we have smooth muscle. The areas that there's a lot of hyaline cartilage, the smooth muscle doesn't do much. So as the hyaline cartilage starts to go away, the smaller and smaller the bronchioles are, you end up with a little more smooth muscle dominated areas. Those are the areas that are affected by asthma, because asthma is when you have the smooth muscle constricting, and it's actually going to constrict the airway slightly. And it can do that more effectively or more easily and have a greater impact on people when their hyaline cartilage is absent, because the hyaline cartilage kind of prevents the airway diameter from changing too much. Then, let's see, surfactant production. We talked about surfactant already. I jumped ahead. So surfactant production prevents collapse of the alveoli. It's a concern only in babies that are born a couple weeks early, because they often don't have enough surfactant. That's known as respiratory distress syndrome for those babies. So they're given an aerosolized version of surfactant so their lungs don't collapse. The pleura is the connective tissue serous membrane around the lungs. We have the parietal pleura that's touching the inside of the chest wall. And then we have the visceral pleura, which is on the surface of the lungs themselves. The relationship of the parietal to the visceral, there's a fluid between, so it slides against each other very, very easily. But because they're essentially stuck together, when the muscles of our chest wall lift our chest wall up and out, the visceral pleura, which is attached to the surface of the lungs, which is also attached to the parietal pleura on the inside of our chest wall, and our diaphragm, when the diaphragm goes down, it's pulling the whole lung downward too because of that relationship. When the chest wall pulls out, it's pulling the lungs outward too. So we breathe by contracting our muscles, first our diaphragm going down and our external intercostals lifting our chest upward. If we want to take a bigger breath, we'll add in the scalenes, sternocleidomastoid, and pectoralis minor as well. If we want to breathe out, the diaphragm just relaxes, but if we really want to force air out, we're going to bend over, we're going to contract our abdominal muscles, and then we have our internal intercostal muscles inside pulling that chest wall down. The lung volumes and capacities, you should be prepared to see a spirogram graph. You should be prepared to know the volume amounts for each of these, whether it's a tidal volume, inspiratory reserve volume, expiratory reserve volume, residual volume, or some capacities, which would be inspiratory capacity, that's your how much you can breathe in plus the inspiratory reserve together, or your functional residual capacity, it's your expiratory reserve volume and your residual volume. The most important one is after you maximally inhale, you fill up your lungs as much as you can. From that point, your maximal exhale that you breathe out and let as much air out till you can't let any more out, that volume of air is known as vital capacity. So you should know about that one. That's about 4,500 milliliters. Factors that are affecting gas diffusion across membranes, that would be partial pressure difference. So you have a whole lot of molecules in the alveoli of oxygen, less in the blood. So that partial pressure difference is going to drive the oxygen out of the alveoli onto the blood. The other factors are going to be membrane thickness. You want a nice thin membrane. A thick membrane is going to impair oxygenation. Then we have surface area. You want as many alveoli spaces interfacing with blood as you can possibly get so we can be more effective at our exchange. Then last but not least would be ventilation, perfusion, distribution, which also leads us into the zones of the lungs. Zone 1, we have a lot of air, a lot less blood. So whatever blood makes it to zone 1 is going to get 100% oxygenated. Zone 2 is going to be the ideal. It's pretty much well matched for air and blood. It kind of comes in these pulsatile surges. Zone 3, we tend to have a lot more blood than the air that gets there can oxygenate. So we don't ever get 100% oxygenated from zone 3. So collectively from the lungs, when we have the oxygenated blood leaving the lungs, it will be about 97%. We never get it to 100% because of the zone distribution, known as ventilation perfusion mismatch. So we just talked about the factors that determine gas exchange. So again, partial pressure difference, membrane thickness, surface area, and ventilation and perfusion matching. You should know basic oxygen partial pressures. In the alveoli, and we're all talking about sea level here, in the alveoli it would be 104 millimeters of mercury. An arterial blood oxygen level would be, say, 100 millimeters of mercury, and that's different than 100%. That's about 97%. So it's 100 millimeters of mercury, and then a venous would be pretty much anything below 80, if you really want to think about, or 60 or below, ideally. Probably is more, but more so you have a ballpark. So if you say somebody has an arterial, value, partial pressure of oxygen at 50 millimeters of mercury, then you immediately go, oh, it's going to be in a vein. You just kind of have an idea where they are. So that's the only, you have alveoli, artery, vein, just these basic ballparks. Oxygen and carbon dioxide transport. Oxygen is transported primarily bound to red blood cells by hemoglobin. Carbon dioxide is primarily transported as carbonic acid in the plasma. What are factors that are going to affect oxygen and hemoglobin binding and unbinding? I find it easier to think of a hot, sweaty, burning energy muscle. So if you want to think increase in temperature is going to help oxygen and hemoglobin unbind. If you have acidity, the presence of acid, we've learned the carbon dioxide, when it's released from the cells, combines with water to become carbonic acid. That by itself helps oxygen unbind from red blood cells. If you're really exercising, you're making lactic acid, you just create even more acid. That's going to help unload even more oxygen because your cells clearly need it. So if you think of increase in temperature and decrease in pH is going to help the unbinding. And therefore warmer or cooler temperatures and more alkaline is going to help facilitate binding. Fetal hemoglobin is slightly different than postnatal hemoglobin in that it binds. The term is known as a higher affinity for oxygen. It's going to bind more avidly to oxygen because it's trying to extract more from mom, the fetus is. The central nervous system control of respiration. I make this a special line item because I want you to realize that it is carbon dioxide also converted as a low pH is what's really the trigger for the pons and medulla in the central nervous system, where it's oxygen that's really affecting the peripheral chemoreceptors in the baroreceptor, or sorry, in the carotid sinus and in the aortic arch. The terminology for oxygen and carbon dioxide, you have hypercapnia, that means a high level of carbon dioxide, hypocapnia, low carbon dioxide. We get hypercapnic if we breathe too slow. It's kind of like holding our breath more. We get hypocapnic if we hyperventilate and like panting, because rapid shallow breaths breathes off. And those, because the carbon dioxide is what's affecting our brain, have a greater impact on control of our diaphragm and our muscles of breathing. So chemoreceptor locations and chemicals detected, that's really back to central nervous system, pons medulla, detecting carbon dioxide or low pH. Peripherally, aortic arch and carotid sinus is primarily going to detect or respond to oxygen.